Hi, my name is Artem Babaian, and today I'm going to be sharing with you my work on petabase scale sequence alignment, catalyzing the discovery of novel coronaviruses. It doesn't take a lot of motivation to convince you that it's important to study coronaviruses right now. The entire global research community has been analyzing now over a million SARS-CoV-2 genomes. And so while the global community is focusing on what's unfolding in front of us, me and a group of collaborators decided, how about we look exactly the other way? Can we understand where this virus came from in evolutionary terms? And this is actually an important problem. SARS-CoV-2 is biochemically predicted to infect over 80 species of mammals. So if this virus enters one of these mammals, it can combine or recombine with some of the native coronaviruses to those mammals. Now, when this recombined virus, say, changes its spike protein and re-enters humans, this actually leads to a very real risk where this virus will be able to reinfect humans, and the herd immunity that we've worked so hard on can be lost, which would pose quite a danger to us. This was just a prediction early on, but now we're seeing that this is actually happening all over the world. So there are tigers that were infected with SARS-CoV-2 in Knoxville and Brooklyn, dogs and cats in Ontario and China, and then a very tragic case of minks in Denmark where the virus infected minks and then went back into humans, leading to the culling of millions of animals. And so we decided, okay, what if we were to just analyze everything, every sequencing data set that contains RNA that we can get our hands on? And very fortuitously, back in February of 2020, NCBI mirrored the public database, the sequence read archive, onto AWS S3 systems. This gives us an unprecedented level of access to massive amounts of data. How big? Well, one petabase is 1 followed by 15 zeros. And currently, there's over 50 petabases, which is roughly equivalent to about 100 petabytes of sequencing data. And this data is growing exponentially. And so while the availability of data is just exploding, outpacing Moore's law even, our ability to access that data in a meaningful way is falling behind very quickly. And this is where cloud computing on AWS can actually really excel. And so this gives us access to massive amounts of data. We're talking every corner of the planet, from cell lines in Vancouver to anal swabs of penguins in Antarctica. All of this data is right here at our fingertips if we can analyze it. And so, we did this as an open collaboration as part of this hackathon, HackSeq-RNA, that happened in Vancouver in May of 2020. Altogether, over 15 of the world's leading researchers came together to tackle this important problem. And so what we came up with was what we're calling the Serratus project, or the Serratus architecture. And the main idea here is to go really, really big, you have to first go small. And so we aggressively cost optimize this architecture to process petabytes of data. It functions natively on AWS and gives us access to over 22,000 virtual CPUs at once. So there's a few kind of key points that I'd like to share with you that will apply to pretty much anyone doing bioinformatics research. So the first thing to consider is that alignment is what is called an embarrassingly parallel problem, meaning that every read pair can be aligned or processed on the CPU independently of every other read pair. And so what we do actually is, instead of trying to work with very big files that can reach, say, 100 million reads per file, we break that up into very uniform blocks. We're calling FASTQ blocks. And we do the work on these FASTQ blocks. Now, the way that I'd like you to think about this is that this creates kind of an atomic unit of work. So when you have a variable file size, you have to essentially engineer your pipeline for the worst case scenario. Otherwise, the big files will fail via errors where there's not enough disk, there's memory issues, or the runtimes are too long. And then if you engineer your pipeline for that, for the kind of the worst case scenario, then you have a lot of slack when you have these smaller files. Whereas if you have a uniform workload, it's very predictable and reliable, the CPU usage, the memory usage that you expect from every kind of atomic block. And this allows you to create a reliable cluster, which overall means that your efficiency will go up significantly. One way to think about this is that it's very quick to optimize for throughput when you have a predictable file size or a predictable unit of work. And so, especially with the smaller instance classes, the amount of networking available to each instance is a fixed amount. And so you can actually swap out the amount of individual threads per node to match the networking input, thus optimizing for I/O. And you can choose to have kind of an optimal amount of disk, memory, and CPU usage based on your applications. So one kind of way to contrast this is if you have something which is more limited by I/O or the ability to move data, you can use smaller instances and you get many of these network inputs. Whereas if you have something that's more CPU limited, you can use one node and then have more CPUs per node. So here, the difference between C5 large and C5 2x large is 4 times the networking speed. And so based on the applications, you can just choose the nodes that work most optimally for your workflow. The other thing to consider is that by creating smaller units of work, you're mitigating the risk of fault in your entire pipeline. So if you have a very large file and you're processing that, if you say get a spot interruption or there's some kind of bug that arises at 60% completion, you lose 60% of that work that you've already kind of paid for. Whereas if you were to break that up into smaller units of work, then smaller units can complete. And that same interruption, say you lose it at 60%, but you only lose 1/4 of the amount of work. And then when you scale this up to a bigger system, this adds up to significant cost savings. So overall, what does this look like? Well, for the alignment modules, we can access just over 4,000 C5x large instances, and we either use Bowtie 2 for nucleotide alignment or Diamond for translated nucleotide alignment. Everything runs on individual threads, which means that we have over 16,000 simultaneous alignments processing all at once. This is moving 100 gigabytes of data a second with an overall CPU usage of about 75%. The way to think about this conceptually is that this is very similar to using a RAID 0-like system, where instead of reading a file once from start to finish, you can actually read a file, say at 100 places at once, thereby increasing or completely bypassing essentially conventional I/O limitations. This is all tracked and monitored with a Grafana and Prometheus interface, which gives us real-time tracking of the cluster's performance. So if any issue arises, we're able to actually bring down just sections of the cluster and fix the issues in real time and then bring them back up. This prevents costly overruns from occurring, thereby saving money again. So what does this mean at the end of the day? Well, using AWS, Serratus can process in excess of 1 million sequencing libraries per day. The cost per library is less than 1 cent. We are talking about massive, truly petabase scale sequence alignment here. And here's just a breakdown of what that looks like. In 24 hours, we analyzed 1.29 million libraries. And if you look at the breakdown of costs, you can see that pretty much the majority of the cost is focused on that computing, which is where you would want your costs to be. By having these small units of work, we're able to essentially eliminate conventional block storage or movement of data that would be associated with that. Really, every step here is optimized to work at a massive scale. So what are the kind of take-home messages of this type of philosophy? One is create a uniform workload, and this gives you very high stability of moving data through your cluster. This also allows you to engineer in spot or fault tolerances, and that allows you to use the much cheaper spot instances for doing this type of analysis. And then again, by using smaller bits of data, you can scale your cluster to really bypass conventional I/O limitations, which are really the bottleneck for trying to analyze a petabyte of data. Go really, really big, but do it by going really small. So what did we actually do biologically with this? So very quickly, we realized that we don't have to just focus on coronaviruses. We actually studied all RNA viruses. So from public sequencing databases, we identified 15,000 species of RNA viruses defined by 90% amino acid identity of the hallmark gene RNA-dependent RNA polymerase. You can think of this as kind of like the heart of an RNA virus. And so we classify these based on their catalytic cores. And using these sequences, we're able to analyze 5.7 million sequencing data sets, or over 10 petabases of sequencing data. You can see that the global distribution of RDRPs covers every corner of the world, the oceans, the mountains, deserts. Where there's sequencing data, there are RNA viruses. And that's actually true for any kind of, say, enzyme that you might be interested in. So what's the punchline here? In 11 days, we found over 130,000 novel species of RNA viruses. To put that into perspective, that increases the total known RNA viruses found over the last 130 years of research by an order of magnitude. And that was done in under 2 weeks. So we're talking about a massive expansion of RNA viruses. And this is purely done by being able to access public data that's sitting there on AWS systems now. So bringing it back to coronaviruses, what does this mean? Well, you do kind of an unbiased analysis to find things that you don't expect. And that's exactly what we found. So we were able to identify 9 new species of coronavirus. And the most interesting ones were associated with aquatic vertebrates. So here we have axolotl, seahorse, pipefish, fugu fish. There was a similar virus identified in Pacific salmon, and these viruses cluster with those. And one thing that was a kind of a paradox and bugged us for the longest time is that we were never able to identify the spike protein on these coronavirus genomes. We'd always get the RDRP segments. And what actually happened was that these viruses appear to be segmented. So they occur on 2 separate molecules. If you think of SARS-CoV-2, the genome is one RNA molecule, but these groups of coronaviruses, they occur likely on 2 different molecules. So if you think of where segmentation also occurs, influenza is another place where this will occur. And so the thing that you should kind of be aware of or think about is that with influenza, these segments can reassort or kind of swap out between different viruses. Now we understand that this could actually also happen in coronaviruses. This doesn't pose any kind of threat to humans, but because these viruses are quite diverged, they infect like axolotl, but it does tell us something very important about the basic biology of coronaviruses. We make this assumption that it always have to be on this one molecule, but that's not true in evolutionary time. And this is kind of the evidence supporting that now. And so, we're rewriting the textbook definition of what a coronavirus is. The other way that we were able to analyze this data is to look for discordant libraries, which is kind of like where you have the coronavirus and then you're finding it in a place that doesn't, you wouldn't expect to find that coronavirus. So we found a bunch of coronaviruses in plant samples, probably because, say like a pig pooped and that was fertilizer for the corn. And so the virus was found on the corn sample. But we had a very interesting case that, again, you don't expect to find these things, but when you do these unbiased searches, you do. There were 2 children sequenced in 2010 who had fever of unknown origin. And within their blood, there were nucleic acids for murine hepatitis virus, which is a beta coronavirus that infects mice. So normally you would not expect a mouse coronavirus to be found in humans, but here we have 2 children where this is the case. And so this actually leads to a very interesting possibility where this type of unbiased metagenomic sequencing can be a very important diagnostic for monitoring for zoonosis or spillover of viruses into humans. And so instead of waiting for this to amplify and affect many people and then be identified, you can identify these spillover events very early on. And then you can act on this information quickly where you can contain it from a public health perspective, essentially immediately before you allow for that kind of exponential growth to kick off. One caveat here is that the presence of nucleic acids do not prove that infection has occurred, so it only is implied. So we don't know if this virus actually infected these individuals, but we know that the nucleic acids for the virus was in their blood. So it's, one of the wonderful things about doing kind of an open science project is that we can also share our data freely. All the data created by the Serratus project is released into the public domain immediately, and it's available on our front-end interface, serratus.io. Here you can search by virus family, virus, and find where any virus, any RNA virus occurs in the SRA, so over 5.7 million samples. And right away, you can just access and find where these interesting cases occur. There's also a R-based interface called Tantalus that gives you programmatic access to this data. So do bioinformaticians dream of Exabase scale sheep? I think I'd like you to come away from this with a completely different perspective on what's possible in bioinformatics today. When you're focusing on on-premises systems, we're really limited by the amount of data we can access at once, the speed and the efficiency that this happens. But if you kind of think cloud-like, this study is kind of like a blueprint for what bioinformatics should be in the next 5 years. We are talking about doing truly planetary scale sequence alignment and study the Earth as a giant sample, not just like a few thousand samples from a cohort or something, but can we study the entire planet from a genetics perspective? Keep in mind, this is in a context of exponentially growing sequence databases. So, what is 5.7 million now will very quickly exceed 100 million samples. And there's no way to access that amount of data in a meaningful way except to leverage cloud-based systems. And so this is, kind of a challenge to all of us, is we should not be building even infrastructure or software to do kind of a petabase scale sequence alignment. We should be thinking, when are we going to analyze an exabase? So I'd like to truly deeply thank all the contributors that have helped make this project possible. This has been a phenomenal project, and it's been an incredible pleasure to work with everyone. I'd like to also give a heartfelt thanks to the UBC AWS Cloud Innovation Center and everyone there who helped make this project possible and helped get it off the ground, and AWS for donating those CPU credits. Thank you all for your time, and I hope that soon we could see each other and have this face-to-face. Take care.